Alright, the impact of yeast presence in nectar on bumblebee behavior and fitness, presented by me. For a little introduction on pollination, about 90% of all plants are pollinated by animals. Most insect pollinators are able to cover large distances, but they require high amounts of carbohydrates. So floral nectar is the main reward plants offer to pollinators, and it is an aqueous solution composing of sugars like sucrose, but it also has amino acids, lipids, organic acids, proteins, and all the other things that they need to survive. So different carbohydrates can offer different terms of like nutritional quality, implying that, relative concentration of sugars and all that means that some have more energy to provide for pollinators than others, and obviously they'd be more attracted to the more efficient nectar. And in terms of like the relationship between nectar and yeast, it's important to understand that like selective forces are exerted by pollinators, so they prefer, certain types of nectar. And recent studies suggest that nectar properties are also impacted by the presence of other organisms, such as yeast. So, there are other microorganisms that can exist in the nectar that change its makeup. And so nectar yeast has been found in lots of plant species, but with only a few taxa. So this suggests that, like pollen, the yeast is being transferred by the visiting insects. And then it also talks about how the yeast depletes the sugar in the nectar, except for fructose, and changes potentially could change like the taste and scent of nectar because of the change in amino acids and stuff. Basically, like what the yeast feeds off of and produces inside the nectar. So like a big question being posed is like, would bees prefer the yeast infected nectar? Would they be more attracted to it, or the opposite? Yeast dependence upon insects for dispersal suggests an interdependence, possibly an intimate relationship between microbes and the insect hosts, kind of like a symbiosis where the yeast provides the host with decomposing enzymes and products. So for a lot of insects, the gut represents an entry route for pathogens and beneficial microorganisms. And yeast cells may become part of that for bumblebees in the gut flora, the microbiota. So it would be important to examine how yeast may benefit preventage of like pathogens or digestion for the bumblebees. And it might improve fitness despite the fact that the yeast in principle reduces the nectar's like carbohydrate benefits. So the hypothesis for this study was basically they're going to examine the impact of nectar with and without yeast on the overall development and health of colonies on a like colony level and individual level, theorizing that the bees will prefer the more nutrient-rich non-yeast option, and that the yeast actually will harm the potential colony like fitness level. But they also propose reasons that it might actually help, like potential benefits of the gut microbiome. This study chose the buff-tailed bumblebee because it's eusocial, endothermic, and requires large amounts of nectar in order to heat themselves and their nest. And they chose the 5 most common species of yeast found in nectar, and they used a sterile sugar water solution where they would inject, like a yeast malt broth, and this yielded 6 treatments in total with different forms of the yeast added, but I'll go into that on the next slide. So 432 queens from a single batch of a rearing colony from Biopest Group were taken from hydrogenation and placed into a plastic nest, and these nets were kept at 29 degrees Celsius and 60% humidity, and it was controlled. And just to avoid minor variations in like the temperature and humidity in the room, the colonies would rotate throughout the experiment. 432 workers were added to the queens after 3 days to facilitate egg laying, and the study was done over 16 weeks. And so for each treatment, 24 replicates were established, and a minimum of 20 was kept out of the experiment because of deaths and such. And for each bee colony, there was 2 containers. One was the control, and the other was the specific treated container. And they were placed underneath the plastic nest, and the sugar solution was made accessible to the nest using a capillary wick. And so you'll notice there was a T1, which was sugar water, just normal inoculated, and then T2 was inactivated after 3 days. The T3 was inactivated cells on a controlled diet. So they just added like inactivated yeast pellets. And then T4 was inactivated after 3 days and then filtered. And so that provided 4 other treatments rather than just like the 5 different yeast species. So in terms of methods and what was tested, they tested changes in nectar chemistry using high-performance liquid chromatography. And basically they just determined like the concentration of 10 sugars, 18 amino acids, and were able to like collect 100 milliliters of the treatment solution from the feeder and analyzed it to understand what yeast did to the overall like composition of the nectar. And then there's also nectar consumption and foraging behavior. Fairly simple. They assessed the nectar consumption and which trays were preferred, and how much was consumed on each to better understand how much nectar the bees needed depending on the treatment. Then there was colony development and fitness. Over the 16-week period, they assessed the larval development time, the timing of the first egg laying, the first development of pupa, first emergence of adults, and after 16 weeks, the colonies were frozen, and they counted the number of males and queens. Health and development at the individual level, pretty self-explanatory, just like assessed the health. And then in terms of mating success, after the 16 weeks, they would take the queens and place 2 males with them with a 1-to-2 ratio, and would determine like how successful they were at mating. And they like determine that based on like who coupled and for how long. And then there's pathogen inhibition in vitro. So basically they would simulate the gut environment inside of the bees, and it would test the ability of a pathogen to grow with non-yeast and then yeast, all 5 species. And in terms of data, I just thought it was important to note that they used all statistical analysis using R for each model. Post hoc analysis of paired comparisons of factor levels were done just by calculating least squared means using the typical tests that we have practiced in this class. And getting into results, the yeast impact on nectar was pretty much exactly what they predicted. There was reduction in amino acid concentrations, and the sugar concentration did change between yeast species, resulting in like a significant increase of certain sugars determined by the certain yeast species. They also identified an increase in fructans, known to have prebiotic effects in animals. So a little bit of a suggestion that maybe there is some benefit in terms of digestion for the bees with yeast. And going further in results in terms of consumption, initially the non-treated sugar water was preferred, but by week 6, the treated sugar water was consumed at a higher rate, and the treatment had impact on consumption preferences, with T3 consistently having lower consumption rates compared to the control. And the data suggests that the suspended yeast was preferred, but that there also wasn't like super significant like differences in foraging behavior. And then so like basically it suggests that bees do not identify and seek the inoculated nectar. The theories surrounding like the different scent or taste doesn't appear to have a significant impact. And in terms of colony development and fitness, actually, it was contrary to the hypothesis. There actually weren't any like significant consequences in terms of like the yeast. There actually was some positive effect on the average number of workers per colony at week 10. But all that, like the time to lay eggs, produce first pupa, rate of colony development, all that pretty much was insignificantly different between the controls and treatment. And in terms of individual mass and size, thorax width was only affected by yeast species in the case of workers, and no difference was found amongst the queens. But some of the yeast species during colony development led to a slightly elevated mass loss in daughter queens. So like when they were frozen and they were like forced to like consume whatever they had inside of them, they had less sugar because of the yeast inside their gut. In terms of mortality, I didn't have any bullet points because the graphs are actually pretty self-evident and easy to explain. All the species of yeast actually led to a decrease in mortality rate, except for mucilaginosa. They were all actually significantly less. So that is an important thing to note, that there is some benefits to the yeast species in terms of mortality rate. And in terms of mating success, pretty much all of the results were fairly insignificant. But mating success did seem to like slightly decrease with a lot of the yeast species. And in terms of pathogen inhibition, the yeast species were added in vitro, and the survival of the pathogen did decrease. But they also found that when they tested the microbiome inside the bumblebees, that the yeast did not actually stay in the gut microbiome, and it was flushed out. So the possible mutually beneficial relationship doesn't appear to be present, at least not terms in microbiota. In terms of conclusion and discussion, overall and contrary to their hypothesis, they concluded that none of the aforementioned yeast species had a negative impact on bumblebee fitness and individual performance. But the species, the yeast species, do vary in impact, and there are some that are more beneficial than others. And in terms of what's next, floral microbes, nectar-inhabiting yeast, have the potential to mediate plant pollinator mutualism, and this effect goes beyond microbial effects on nectar chemistry and sensorial cues, such as the presence of cells or volatile production. So basically what they're saying is there is information that we're not quite sure why it, because in theory the yeast species should reduce the fitness, but they don't. And so more studies need to be done in terms of why.